half of the people already joined in. Can you ask your friends to wake up? I know this is the first day for the semester. If we start early, we can finish early. I have just reminding you, I have put the copy of lecture today on the Putra Blast system for both group. Okay, for group number 1 and 3. So you can have that. If for some reason you cannot view it, you need to let me know. Can I appoint someone to let people in while I'm teaching? I'm afraid that some people might come a bit late and then I cannot stop to let you in. Probably I should have used my regular Google Meet. They're already turned on. I'm going to log out for 1 moment because I just installed an extension on the Google Chrome to let people going in automatically. I'm not leaving, okay? I'm just, I'll come back in a bit. Okay. Yeah. Let's see. This thing for some reason it doesn't allow you guys to join in automatically because I'm using the UPM server now, and it detects you from outside of the server. That's not very friendly. Not to worry. Oh, while we are waiting for your friend to come in, can somebody, or can we appoint someone to be the class representative for each group, so that you can represent your other friends to communicate with me later? Anybody wants to volunteer, or anybody wants to suggest anybody? Have you been in my class? No, right. Oh, who created the WhatsApp group? That was group number 1, right? Whoever that is. Are you here? Okay, I think we should make a start. While waiting for your friend, I think I'm going to share my screen now. Let's see. Oh, can you see my screen? This thing is not telling me whether are you seeing my screen or are you seeing something else?
Not yet, doctor.
Not yet.
Not
Good.
yet, doctor.
Not yet. Okay, I'm going to share my screen now. Screen. That's not it. Okay, what about now? Can you see my screen?
Yes, doctor.
Yes, doctor.
Okay, good. Thank you. All right. So, welcome to Crop Physiology, AGR 3301 for group number 1 and group number 3. I want to let you in just one group, but the system doesn't allow it for the lab sessions. The system allows for the lecture. But it's all right. I'm sorry if you get disappointed for not being able in group Dr. Putri, but she is not able to teach you for this semester. She got just way too many things to teach apparently. Okay, so let me open something so that you can see teaching plan. Yeah. So that's the teaching plan. Let's do this. Okay, I'm going to open. So I'm just going to give you a brief outline of what's going to happen for this semester. So this is the teaching plan, the lecture plan, and the practical plan from last year. But this is pretty much what we're going to follow. I can give you a copy for this later. I just need to change the dates and make other corresponding things correct. Okay, so you're gonna have your lecture and practical, and for that, your lecture will take place on Monday, today, 8 until 10. For group 3, on system, on your ESMP system or Putra Blast system, it's going to say that your lecture is at 2 p.m. later today, Monday, the same day. But do not go to any class at 2 p.m. I'm not going to be there to teach you. Come to your this time, okay? Your original time. We just changed the system for a bit to allow my name to become your lecturer simultaneously for 2 groups. So we're gonna, for your lecture, this is pretty much what you're gonna have. So we're going to start with a very simple, very a mild introduction to the plant physiology. What was that all about from the point of view of agriculturist and horticulturist. Okay, and then correspondingly we are going to do the same lab. Okay, practical activity which is going to correspond for week 1 here and week 1 there. I think this practical plan for the lab has been slightly changed. I'm going to check with our lab assistant later, but I don't think it's going to be very far from here, especially for the experiment for the growth analysis. I think we're going to start the experiment this week because we need to grow some plants. Yeah, it's going to take some time for the seedling to germinate. So we're going to start this week, and we're going to see the results in the coming weeks. Okay. Okay, so for the lecture, we're going to have all these things: seed, germination, dormancy, photosynthesis, and the rest of it until your mid-semester break, okay, which is right in the middle of the semester. And then we're going to continue, and you're going to see that we're going to have this class discussion case study. These are the things which are flexible. Why do we have this? Because your maybe I can show here. Doesn't see here. Yeah. Yeah, this is pretty much how we're going to evaluate you. You're going to have various things evaluated based on your knowledge, your skill, your communication prowess, and then your long life learning capability. Okay, so I'm going to use this scheme. So the uh, allotment for your scoring would be something like 60 or 70% based on the coursework, including your test 1, test 2, presentation, case study, and things like that. And then the remaining 30 or 40% for your final. Okay, so there's a lot of room to help you to get good marks in this course. Okay, so practical. We're going to do the practical face to face. You need to come, but do not cramp in one lab. Okay, if you're group number 3, just go to your original planning for group number 3. Okay, I will try to set up a system so that if I'm giving the briefing for group number 1, you on the people next door can know as well. Okay. For that, I think, yeah, that's pretty much it. Is everything clear so far? Are you in the right class? Make sure you are in the right class. Okay, just want to check the message here. Okay, cool, thank you. So just follow this, okay? I'm going to start the lecture now, so I might not be able to admit anybody else anymore. So I give 1 minute to ask your friend to wake up and join. And then you can, crash back to your bed if that's what you do. So ask your friend. I'm going to start in 1 minute time. Thank you. Somebody send a chat. What does it say? How to turn on the chat for this thing? Okay. So you got 10 days until 27. Why? What happened? Yeah, did you come late? I see. Okay. I'm going to write down your name because I need to let our coordinator know. Can you tell that's your name? Can you give me your metric number as well? You know what? We're going to do this at the end of the lecture. Why not anybody of you among the 60 of you, if you cannot come for some reason to the lab sessions this week, let me know. Some of you might get quarantined. Some of you, got other lively issues, right? Okay, right. And with that, let's start with the lecture. Yay. Okay, where's my pen? I'm going to turn off this. This is not helping, actually. Okay. So welcome to Crop Physiology 3301. And for those who have not seen physiology uh, before, the word itself, not to worry. Um, this is why you're here, okay? And it basically deals with something inside the process and the mechanism inside the plant, and that's what you want to know about. Okay, so for today's lecture, I'm going to introduce you to the subject itself, what it's all about, and what's gonna be learned from it, the importance of it, why is it very important for agriculture student to learn. And then we're going to move for a bit to the crop growth analysis, which is pretty much a classic thing to do, but it's still relevant until today. Okay, so as you can see, we got two sections for this lecture. Okay, all right. So so before you go deeper into the subject, we would like to know, is it crop or is it plant? I know that many book do not call this as crop physiology, but rather call it plant physiology. Okay, we got the crop, we got the plant. The first you need to distinguish between what is regarded as crop and plant. Okay, the most basic thing is you need to know all crops are plants, but not all plants are crops. Crops are actually plants which have been commercially grown. So it involves the elements of profits, subsistence, and also it sounds very lucrative. Okay, so that's why it's more relevant to use the word crop for agriculture and horticulture. Okay, so is somebody going to call you, oh, I got some crop at my lawn, at my home. So you're going to wonder, do you sell the crops? No. If it's not, if nothing commercial about it, no, you just call them plants. Okay. So regardless of the crops that you grow, you want to focus on the certain organs of the crops, of the plants. Okay, so some crops you are interested with the leaves, some crops you are interested with the seeds. Okay, so this is another good differentiation between the crop and the plants. Okay, you have your organ of interest. Okay, sometimes you harvest these all. Okay, but for the plants, you admire the plant as the entity as a whole, rather than focusing on certain harvest organs. Okay, so why is it important then to understand crop physiology? The physiology itself is talking about the process, the mechanism that is actually happening inside living organisms. Okay, so it concerns with the process and also the function. So these two things are brought together, and then you have the discipline, the subdiscipline under plant science, which is called crop physiology. Okay, and so to give you some basic understanding of the process, so process are actually the natural event or the sequence of events that even without you telling the organism to do or stimulate, the organism are going to do that anyway. Okay, so we're talking about the plant, of course. So you're going to have all of these which are relevant. We got the photosynthesis, ion absorption, transpiration, assimilation, stomatal opening, and so on, because you are dealing with crop or plant physiology. So these are the common processes that always take place. But when you're talking about human physiology, if you are medic students, you're not going to talk about photosynthesis. Okay, medic students do not have to learn photosynthesis, but they have to learn something else. Maybe they have to learn about vitamin D formation. Okay, vitamin D synthesis from you know, when you are exposing your skin to the sunshine, there's some chemical reactions going to take place. So for the human physiology, that's what you're going to learn. And also there are physiology in other living organism. We got the insects physiology, we got the bacteria physiology, and also the fungus physiology, as long as they're living. Okay, and then we go a bit after you have understood there are many processes inside a living organism. These processes on its own or collectively can carry out a function. Okay, there are many functions. For example, I put it over here. Plant can functions to capture energy and assimilate carbon. This one function, energy capturing and carbon assimilating, even though it sounds like only one function, but in order to accomplish this, two processes actually take place. Number one is the photosynthesis. Number two is the cellular respiration. Okay, so for a function to happen, maybe one or more process need to take place. Okay, and also the other example of plant functions, including the nutrient and water distribution, growing and developing, and so on. Okay, so in essence, the collection of processes, like what you have seen here, will give rise to a function inside a plant. So you can only imagine now how complex a plant is. It can be very um, daunting for you to understand somehow, but that is what is taking place. I mean, like, even for you, I mean, like, it's not like you are plugged into to a wall and move about. Okay, we are pretty much like wireless, you know, because we have our own uh, energy reactor inside us that derive the energy from the food and enable us to do all these amazing things. So pretty much like that for the plant. We're not like machine. Okay, so I would like to remind you as well about the things that you need to recall. I hope you have not thrown away your botany lesson just yet, because you're going to use that a lot now. Okay, so this is a typical representation of a plant. Okay, what you need to know, a plant, when talking about the underground and aerial part of it, comprising shoot system and the root system. Okay, root system is anything below ground. So you got the root, you got the root hairs, and maybe you know, all the mycorrhiza and all the bacteria that may be becoming part of the root. Okay, and then you have the shoot system. The common misunderstanding here is people always think shoot system only this part. No, shoot system comprises whatever from the base of the plant, starting from the ground, all the way to the top of the plant. So you're gonna have your the leaf organs, you're gonna have your flower organs, you're gonna have your stem organ, and also you're gonna have your fruit organs. So these organs collectively belong to the shoot system. Okay, which is very contrasting to the root system. The only primary organs here are only the root. Okay, and some plants they maybe have a bit more. They got the nodules. Okay, the nodule, which is part of the root system. Okay, nodule is the specialized structure for the bean family. Okay, to understand the concept of growth analysis, which is the second part of this lecture, we need to get some terminology correct. Okay, because people keep on having this misconception, and then when you misunderstand it, of course you're going to use it wrongly. So let's get this correct and have it embedded in your mind, because you're going to use this for the rest of the time, as long as you are still an agriculture enthusiast. These are the things that you're going to see all the time. They are the growth, development, and differentiation. Okay, if you use the Malay, I think this is the pembesaran, perkembangan, dan juga pembezaan. Okay, let's start with growth. Number one, growth is something, is a process which is irreversible, meaning that there is no U-turn. Once it has taken place, that's it. Okay, it involves in the event of cell division and enlargement. Okay, the cell has to divide and then it becomes enlarged. So under this cell division, this is where you're going to get your popular mitosis and meiosis. Okay, meiosis. Okay, and then the concept is very simple. I think all of you know, from one cell, okay, you're gonna get two cell, and then from two cell, you're gonna get your four cell. Okay, so when this happens, this what we call as cell division, and this cell, once it has divided, instead of staying this size, it can undergo expansion or enlargement. It can become suddenly longer, so it becomes elongated, or it can grow just symmetrically, becoming a bigger circle. Okay, depending on the type of the cell. Okay, so to put things into perspective, when you look at the plant as a one organism, the growth can thus be determinate or indeterminate. So determinate growth is when the organ or part of the organism reach a certain size and then stops growing. Okay, it means it's determinate, it's fixed. So I've got a very simple example here. We've got two varieties of tomatoes here. So the one on the right is actually the determinate one. You have your plant up to a certain size, and that's it. It's not going to grow any further. And then usually once you cut it, they're going to die pretty much. Okay, because it's not like they can grow new shoots to replenish or to replace what has been lost. Compared to indeterminate growth, for another type of tomato, the vine tomato here, which can pretty much grow in all directions. Okay, so this kind of growth is very rapid and very active. So usually this kind of tomato, we have to put it on a support structure. Okay, this tomato, you can do the pruning or trimming of the plants. Okay, and you're gonna get your fruits. The good thing is you're gonna have it throughout the seasons. Okay, usually it takes longer for the indeterminate tomato to produce any yield or harvest compared to the determinate one. So it depends. Are you looking for the long run kind of harvest, or are you looking for a quick harvest? Right? Okay. About the development. Okay, you have understood that growth is something to do about the increment and size. Okay, from something smaller becomes something bigger. It does not involve the change in the form of physical development. Simply put, it's approaching maturation. Okay, it develops into something else under the study of morphogenesis. Okay, when you see this word morphogenesis, so morpho, it means form. Okay, so we got the genesis here. Genesis, the formation of. Okay, so once the cell have become bigger, it will start to mature. And the process of maturation, this differentiation and morphogenesis will take place. I've got a simple example here as well. We use tomato again. These all start as the same cells. Okay, they're pretty much the same cells, and then the cells become bigger, as you can see from this the phase of ovary development. And from the same population of cells, suddenly physically you get something else. You're gonna get the flower, and then the flower will undergo further cell division and cell differentiation, and through the action of fertilization, you're gonna get your fruit. Okay, your fruit gonna have the cell expansion, and also they're going to develop into something which is completely different from the beginning. You see, when the beginning is different than the ending, you know that the development has taken place. Okay, so quick recap for you: growth, size increment, development is the evolution, right? Okay. The third terminology is differentiation, or in bahasa we call it pembezaan. It's the process of a cell to become specialized, or rather than a generalized cells. So cells in your body or in plants, they can be regarded as general cells, meaning that they do not have any fate or destiny yet. Okay, so this group of cell which kind of look the same and do the same thing. So we call this cell as meristem. They do not have any fate yet, but the moment they have um, uh, destiny laid upon them, they're going to differentiate and specialize. So they will become a special cell. Okay, I've got one good example here. This is the formation of a stoma. You know, you got stomata on the surface of your leaf, the little pores that allow gas exchange for the leaf to do photosynthesis. So this is the formation of a stoma, and this is the stoma for a rice plant. Okay, there are many stages here. So everything starts as the meristem. MMC stands for meristematic mother cell. This is the original cell, pretty much like this guy here. It looks simple, it looks boring, nothing interesting happening just yet. So on the surface of the leaf, if you look under microscope, you're gonna have this protodermal file of cells here. Okay, then due to special sets of gene expression and with the progression of time, um, cell division will take place. Okay, I just want to highlight a small thing here. This is a special place in plant where you're going to get this asymmetric division. Most of the time when you learned previously about your mitosis, you're going to look at the pretty diagram that you learned from school. You're going to get at the end of mitosis two equivalent cells, right? This is what you get. Yeah, this is the regular mitosis. However, for some cells in plant or in human, asymmetric division occurs, meaning that if the original cell look like this, the new cell after division occurs going to get uh, something completely different. Okay, so this is what we call as symmetric division. So after this asymmetric division, more cells will be produced. Okay, for this case. Okay, until it becomes a new set of cells. So at the beginning, you got meristematic mother cells, and now it has transitioned into GMC. GMC stands for gut mother cells. Okay, gut. Okay, gut. This thing here. This is gut cell number 1. This is gut cell number 2. The gut cell. The pair of gut cell actually form one stoma. Stoma singular, stomata plural. Remember that, okay? And then this one GMC, gut mother cell, will undergo the regular mitosis that you learn in school to produce a pair of gut cells like this. And then they're going to form a small aperture between them, and that's how you get your stoma. Okay, so from the beginning of meristematic cells all the way to stoma, these things they developed, they grow, and then they differentiate. You got everything in one go here. So this is a very good example. Okay, so get the terminologies correct, and you will have no problem in remembering that. So for the rest of the time. Okay, so I think we're done with the first part of the lecture, which is the introduction. Also getting your terminologies correct. Any question up to this point? I'm just going to check if anybody tried to come in. No? Okay, good. Any questions so far? Are you good? Still alive? Okay, good, good. Okay, I think we can continue for now. To turn off this. So now we're going to learn about the crop growth analysis. Growth analysis. We want from this word, you already know that we are trying to learn to quantify how big, how fast your crop has become, because it's a living organism. One day it's small, another day it's going to get bigger. Okay, we want to understand how to quantify and how to interpret this increment in size, or maybe the other changes that is occurring for your crop plants. Okay. Again, I want to recall to all of you about your previous botany lessons. Okay, plant growth stages. So these are like a life cycle of a plant. Okay, we've got a tomato here again. For some reason, my examples today full of tomatoes. So we got your tomato start as a seed here, and then it's going to sprout and germinate, and then you're going to get your babies tomato seedling. Okay, after this stage, you're going to get your vegetative state. Okay, this is where the plant is going to actively producing the leaf organs. Okay, and this vegetative state depends on the species. Some species, it's only around 2 or 3 weeks time. Some species can be months, some species can be years. Okay, although the sprouting can be very quick for most of the plant, but the vegetative stage or the seedling stage, and for the rest of the stages, in fact, can vary in time depending on the species. Okay, and actually bear in mind, vegetative scientists sometimes even divide it a bit down further. So you got your early vegetative, and then you got your late vegetative. Okay, so in this early vegetative, where you're going to see in some textbook, they call it as juvenile stage. Okay, nothing to do with crimes. It's just the way it's called. Um, Somebody is knocking on the door. Just want to, I want you to let you in. Why are you late? Keep sleeping. Okay, and then you're going to, after the vegetative state, you're going to move a bit to the budding stage, where you're going to get your leaf organs still there, but now you got your flower buds. Yeah, so this is the time when your plant's going to look so pretty. Okay, and then the flowering stage, and this is the stage actually where the pollination going to take place, and then you're going to get your fruit. Okay, and the fruit inside of it is where you're going to get your seeds, and the seeds here, if you sow it again, this cycle will continue. Okay, so bear in mind, this is the stage, because we have maybe different ways to quantify the growth for each of this stage. Okay. Okay, look at this statement, and also a point of review for what you just learned a few minutes back regarding growth and development. When somebody sow a seed in your garden, okay, and then the seed is going to sprout and germinate, then you're going to get you know, plants, your seedling plants, your juvenile plants, and then it's going to grow and then becomes mature, and then eventually it's going to produce the flower and fruits. Collectively, this growth and development has happened. Actually, I should put one more: growth, development, and differentiation. One more thing. So all these three things that you have learned a while back actually have happened when a plant has completed this entire life cycle. Okay, that's why it's very important for you to get the terminologies correct. And this is another example for the plant. Some plants are going to look completely different. Like for the rice, it looks pretty much the same. Okay, like if you compare during the seedling stage compared to the mature stage, they kind of look the same. But some plant can look completely different. Okay, it's just the genetic capacity for each organism. Okay. So growth, as you recall from before, is the irreversible increase in mass. And how do you achieve this growth? Because the plant has a built-in process to manufacture its own food. Okay, I hope you still remember your little photosynthesis process. So plants can photosynthesize, meaning that they can manufacture their own food. When this happens, of course, they can grow very easily. Okay, so when we're talking about the growth itself is a process. Remember from the earlier of the lecture, you got process, you got functions. This is the process. And under this process of growth, there are sub phases. There are small phases that happen. We're going to have a look at that now. So it starts with the cell division. Cell division with your mitosis or meiosis. And for the example that I have here, this is a root hair, a piece of root. So you're going to, this is the root structure. So you got your cell division phase, which is in this region here. This region here, cell division, where your cells actively dividing, and then you're going to get your cell enlargement, the elongation phase, which is this phase here. Okay, bear in mind, up to this stage, these cells, even though they are different in shape, they are still the same. They are still one type of cells. They just divide and elongate. Okay, they still do not have any specific fate or destiny, not until the cell differentiation or maturation phase kicks in. And further up, that is where you're going to get different root cells. You're going to get your vascular bundle cells, you're going to get the cortical cell, epidermal cell, and also the root hair cells. Okay, so everything happened within this one diagram. So you can understand that the stages of cellular growth is a continuous process, and it happens gradually. Okay, it's not like one at one second it's dividing, and another second it becomes differentiated into something else. No, it's a gradual process. Okay. Um, One thing I would like you to pay attention to, look at this, because this can become a trick question. If somebody asks you, where is the youngest region inside this cell, and label it, where would you label it? For this kind of thing, never label this part, the root cap. Okay, root cap is never the youngest part of it. The youngest part of it is this thing, the apical meristem. This is when the cell age is 0. The root cap has been produced much earlier than this apical meristem. Okay, so for this case, the lowest is the youngest, does not apply. Okay, the apical meristem is actually inside. Root cap actually is a protective cover at the tip of your root, because your root has to travel inside the soil, and the soil is very coarse, and we've got some bacteria and some other stuff, so it needs to protect all these delicate cells inside. So that's why it's got this root cap, and also to kind of poke, making way for the root to travel. Okay, so remember that. And if I ask for the oldest, well, that's a trick question as well. It can be this part which is the oldest, or this part which is the oldest. Okay, but never other parts. So bear that in mind. Okay, so with the cell proliferation, you're going to get lots of new cell. So cell proliferation is pretty much the same as the cell division. Okay, so from one cell, they got 2 cell, got 4 cell, and so on. Okay, and then you got your cell enlargement, and then the growth by accretion. Accretion is actually the growth by combining cells into layers. Okay, so you have maybe you have a one layer of cells, and then they divide, right? They divide like this, and then the cells kind of secrete some kind of special material like a cement, so that the cell on top of it can stick, and now they become bigger and look like one, but actually they have been joined together, cemented together. So that process, we call it cell accretion, the adding of cell by layer. Okay, we have looked at the concept of cellular growth, the phases that they involve. What about, to put it into perspective, when the growth is happening throughout the life of a plant, how would it look like? So in here, this is what we call as the growth curve. So the growth curve, you got your axis here, you got your x-axis down here, and then you got your y-axis on the vertical side here. On the x-axis, you got the progression of time. It can be days, it can be weeks, it can be years. Any way you want to put it on the time scale. And then on the y-axis, you're going to get these growth measurements. So the growth can be measured in many ways. It can be measured in the form of size, weight of your organism. So when you plot this kind of observation using your plant, this is what usually you're going to get. You're going to get this growth curve, or sometimes people call it as a sigmoid curve, because it looks like an S. gonna start it. You can, the way you interpret it is you can divide it into um, 3 phases. I'm sorry if my drawing is not that smooth because I'm not an artist. Okay, so you're going to start with the lag phase. This is the period when the growth rate is very slow because this is the initial stage of the growth. The cell is just waking up. Okay, it's like you when you just woken up from your bed. It's not like you can just go sprint 200 meters. Okay, you're gonna get some time to wake up. One, it has awakened up, it has gathered all the food and energy. This is the log period, the exponential phase. Suddenly you're gonna get all this size, weight, or anything that you measure in terms of growth spiking up. Okay, so if you want to translate this into the phases of your plant, remember this. I will oh, sorry, this thing. So this would be something like your the lag phase. Okay, and then suddenly when the plant spiking up in terms of the growth, this is your exponential phase. And then this would be the third phase, the stationary phase, when your plant's no longer growing. Okay, right. Yeah. So at this stationary phase, usually this is when your plant is ripening, or if it's not an annual, it's oh sorry, if it's not a perennial, it's going to undergo the senescent period. Okay, everything becomes static, and the plant just becoming weaker and more fragile. Okay, this is the aging process of it. A good thing to ponder: can this be applicable to your own growth? Yeah, this would be your a baby phase, and this would be your teenagers phase. Okay, when you are in your primary and secondary school, and this is when you are in your adult phase. Okay, pretty much. I mean, like, are you still growing now? If you will not stop growing, you can be as tall as like a coconut tree. Okay, at one point you just stop. Okay, and then you're going to enter the senescent period. Okay, and hopefully it's not going to happen anytime soon for you, but yeah, that's where we are all heading. Okay, how to measure growth? Okay, to make something pretty like this, how do you want to measure your growth? So in terms of the uh, length of something, we actually, oh sorry, I should put it this way. Depending on what organs you want to measure. So you have your plants. Okay, you have, remember, you have your shoot part, you have your root part. Depending on the organs that you're talking about, this will determine whether you want to measure using your ruler, to see the growth, or you want to measure the area or the volume of it. Okay, so for stem and root, usually we measure the length, the growth. I mean, the growth is usually in the form of weight here. Yeah. For the leaf, usually we go for the area. Sometimes you can use both. Okay, for example, if you take leaf, it's your regular leaf, can you measure weight? Yes. Can you measure area? Yes. Can you measure volume? Yes, you can. There's a equipment to do that. Can you measure length? Yes. Yeah, depending on all. Sometimes you can use all of these for one organ. Sometimes it's not possible. Like the fruit or something. Sometimes it's not possible to know the length of fruit. Like if the fruit looks irregular somehow, like the durian yet, right? It can be very irregular fruit. Sometimes it looks oblong, it looks round, even I'm talking about the same variety. Yeah. So maybe people go for the weight. Okay, so depending on the objective of your experiment, that will decide what kind of way to measure the growth for your organ of interest. Okay, so this is a more detailed way to look at how growth can be measured. You can measure it using the fresh weight. Simply, you pick your organ of interest, your harvest organ, and then weigh them. Uh, However, this thing, even though it's very advantageous, it's very quick and very easy. You just, for example, you got your leaf, you just pick your leaf and put it on the weighing scale, and then you got your result. This is prone to high variation. You see, the same leaf, if you pick in the morning, the weight is not going to be the same if you pick it up right in the afternoon when it's very hot. Okay, the weight is going to differ. Some amount of variation happens. Okay, so there are some disadvantages, and sometimes along with your organs, you're gonna get your dirt and particles because it's a fresh, direct from the farm. Thing, other unwanted things can follow as well. And then to overcome that, usually this is what people do. People do the dry weight. If they need to wash the plants, the organs, they will wash the plants and organs, and then they're going to dry them. Usually about 2 or 3 days, depending on the organs, whether it's big or thick or something like that. Around 80 degree. The temperature here can vary, okay? 70 to 80 degree. It actually starting from 60 to 80 degree. Why there is a difference in the temperature when you are weighing your sample? Because some plant materials, like the leaves of plants which are aromatic leaves, like the mint, like the rose petals, if you subject these things to dry at very high temperature, you're going to lose all the volatile compounds of the leaf. Okay, because drying, the goal of drying is just to eliminate water so that your the actual biomass of your organs without the presence of water. Okay, but if you subject it to a high temperature, you might lose all these volatile compounds, and of course your reading wouldn't be very correct anymore. Okay, so this depends on the species. Sometimes you only have to put like at 40 degree if the organ or the plants is very delicate. Okay, so this is very good, but of course it takes time, and you need to use a more expensive equipment. Okay, longer. You need to accomplish every several days, and then you need to have an oven. Okay, and then you got the length. Okay, this is good if you want to measure the plants continuously, or you are dealing with plants which you cannot destroy. Okay, nothing destructive can take place. Fresh weight and dry weight is very destructive. Once you have plucked your plants, and that's it. It's not like you can put it back to plants. Okay, so sometimes people measure length, maybe on a weekly basis, to see whether the plant is growing to a certain degree. And a length include like the height measurement, the width measurement, the girth measurement, and so on. And you can use a relatively cheap equipment to accomplish this as well. So you can just use your things like um, a ruler. So ruler can go for the length or width, the dimension of it. Okay, and then you can use your caliper. Okay, maybe you want to measure the girth. Okay, and so on, and also the area. Area is usually we use for the leaf. This is quite special for the leaf actually. Okay. we have a machine to facilitate a scientist to measure the leaf area. We just pick all the leaf, many leaf, and then we put it into this machine. This machine got like a conveyor belt. You know, if you look at the documentary of a plant production system, you got this moving conveyor belt. Yeah, we got this machine here where you put your leaf. So the leaf is going to move, and there's a camera inside this machine. The camera is going to capture this leaf and translate it into um, leaf area. Okay, so that's very easy to do. Okay, the growth analysis. There are a few ways to do it. I mean, like, these are the methods to sample your crop or your plants. But after you have obtained all of this data, the fresh weight, the dry weight, the area, and so on, what are you going to do this? You have to put this information into some kind of computation so that some interpretation can come out from your observation. Okay, in order to do that, we use the growth analysis. Okay, it's just a mathematical expression, like formula. Yeah, formula, something add by something, divide by something else, or something divide by something else, and to the power of 2. Yes, things like that. Yeah. Remember equation? You know, like the Einstein famous equation, E equals to mc squared. So in order to get the reading of energy, you need to multiply m. M stands for what? The mass, and c is the speed of light. Yeah. So this is a mathematical expression like this one. So growth analysis can have many equations in order to quantify, depending on what kind of data that you have. Okay. Okay. Why we need to do growth analysis? Because spatially and temporally, we can understand how the plant grow and develop. Spatial means space. Temporally means time. Okay. In space, in a geographic location, specific location, we want to understand how your crop respond. And also over the time, given that it has been in a specific space or condition, we want to see over time how your plants are going to respond. Okay, that's why growth analysis is very important. And then we're going to have a look at the dry matter accumulation. Dry matter meaning that no water, just other elements, because water fluctuates. That's why we need to eliminate water. The things which are not eliminated during drying is carbon. Okay, carbon and other minerals, of course. So these pretty much stay put after the drying process have taken place. Okay. So you can get a very good understanding about your plant performance throughout its life. Okay, and then you're gonna see about the environmental influence and also the genotypic differences between your cultivars. Genotypic means your genetic capacity of your plant, and then after having that specific capacity, how is that respond to the environment? And this is what we call as G by E factor. How genetic interact with an environment to produce your observed plant performance. Okay, and this is going to be different from variety to variety, or from experiment to experiment. Okay, and of course, the growth analysis, this classic growth analysis, this is very classic, okay, because this has been around since forever. It is destructive. Okay, you have to kill your plant in order to get the dry weight. There are many parameters. Parameters are the things that you measure. for the growth analysis. There are many, but we're just going to focus on 5 because I think these are the most basic. And if you have a good understanding of it, you can have something more complex and sophisticated. Okay, we start with the absolute growth rate. You see, it has been around since the beginning of the 20th century, long time ago, almost 100 years ago. So it shows you the increase of total dry weight per plant. This is the simplest one. Okay, you just have the dry weight of a total area of plant between two dates. Okay, so this is the formula. The formula here: absolute growth rate, weight, the final weight of your plant minus by the weight of your plant in the initial period. Maybe W2 is your plant at harvest, and maybe weight number 1 is at the beginning, maybe at its juvenile stage, and then you divide by the corresponding time. Okay, so you're gonna get your formula here, plug in everything correspondingly. You're gonna get in the unit of gram dry matter per day. You want to see the accumulation of dry matter, the carbon, especially the carbon and other minerals, for every single day. So if you put it into perspective in the reality, so this is what the graph is going to look like. Okay, gram plant per day, days after transplantation. So you can see that we can take example, the stem. The number of stem is kind of increasing, and then maybe go up, and then go down again, and then go up again. So depending on the organs, it's going to differ throughout the time. Okay, so you can apply this to a corresponding organ. Okay, like in this graph here. Okay, so very simple, that one. And then the second one is the crop growth rate. Okay, it simply tells you the increase of dry matter per grams per unit area of time. Okay, it deals with the matter accumulation over a period of time. You want to know how good your plant at hoarding all this carbon for a given time. Okay, this is when you're going to see sorry. So what's the difference in between this with the first formula? So you got your formula here, and then you got your formula here. So what's the difference? Actually, this is something that we can discuss in the lab time. But for the time being, I just want to introduce you to this the concept of growth analysis. Okay, so you're gonna get this. Okay, and then you just plug in and you do that like the first time. You're gonna get your crop growth rate. Okay. And then you're gonna get also the third way to do this, which is the relative growth rate. Okay, it has been around for quite some time as well. So look at the unit. This deals with the relative increase in dry matter per unit dry matter. So the good thing about using this RGR compared to the previous parameters is it can be independent from the size of your plant, because we use a special tool in mathematics to standardize the way your data turn out. I mean, like if you compare from maybe you have different people doing things at different time or different places, maybe they're going to look at different organs, even though they're dealing with the same plants, but we are still trying to understand how these plants behave in a very standard way. So we use RGR, relative growth rate. I'm going to show you why. So this is the formula for RGR. Um, Pay attention to this ln here. Um, I hope you still remember your mathematics when you learn. So ln is the natural logarithm, natural log. Okay, you use natural log to transform your data in the equation, so that it can give you something like this. Okay, if you don't transform your data, you're going to get pretty much like what you have seen previously. Okay, you're just going to get a straight line, line up, line go down. You're not going to get something standardized that you can compare. I mean, like if you're trying to compare between two organs, they're going to look like completely different. You can't really interpret it. But if you use this formula, you can turn your observation into the sigmoid curve, like this. Okay, then only you can interpret it. You can, oh, up to this day, your plant is undergoing the lag phase, and then from this period, maybe from week 3 until week 7, it is in the log phase, and then from this week 8 until week 11, maybe it's in the stationary, so you know that your plant maybe is maturing, not producing any more new leaves, no more vegetative state. And then suddenly, if you have something down, you know that your plant is dying. So this is a more meaningful way to do it. Okay. Just to recall you about the natural log. Natural log is usually indicated by the E on your calculator. If you take your calculator, it's either ln or E. Okay, E, this is the value of the natural log. It's always the same. It's pretty much like pi. So pi equivalent to 3.14. So for E, it's equivalent for 2.718. E stands for what? Okay, there are many stories to this E. Some people call it Euler number. Some people call exponential number. It's just a special magic number for the natural log. Okay, remember you learn your log. So you got your log. If you don't say anything, you know this is a log 10. And then if you log E, you know this is actually 2.71. Okay. All right. The next growth parameters is the net assimilation rate. Okay. So this is involving photosynthesis. Okay. So your plants photosynthesize, and we want to see the product, how much of it going to your plant material. Okay, involving the carbon assimilation. Okay, so it tells you about, it tells you about the rate of increase in dry matter per unit of leaf area. So your leaf area is the factory of your plant. This is the organ that do all the photosynthesis. So you want to see if your plant have a given area of leaf, how much of it can have impact on, let's say, your other organs, let's say your stem. Okay, because stem cannot do photosynthesis on its own. It still rely on the leaf. Okay, so leaf is very important. Okay, so this is the formula for your net assimilation rate. Okay, not to worry too much about this formula. If the exam, we're going to give the formula, and in fact I will teach you later a small software to plug in and to get all of this. Okay, so not to worry. So this is the formula for the net assimilation rate, and for the actual use of it, if you plot your graph, this is what you're going to get. Okay, net assimilation rate, this is the unit. Gram of your organ of interest becoming more or less per given area per day. And this is your days after planting. Okay, and this is of different treatment. Okay, net assimilation rate in different irrigation levels, 100% all the way to 50%, and humic acid application. So for example, we can take a moment and appreciate this graph here. So this solid black line that you see here, the treatment is 100% of irrigation with using humic acid H1, the type of humic acid. So you're going to see that at the beginning, the net assimilation is not the highest, but around 30 days or so, it's spiking up for a bit more than any other treatments, and then it's going to come down. And then right around this stage here, maybe around 90 days after planting, the net assimilation rate pretty much like the same for other treatments. Okay, if you compare to this treatment here with the bigger dash, 50% of irrigation level, and then no humic acid. So you're going to see that the net assimilation rate is pretty much the same throughout the time. Yeah, there is no spiking, everything stays consistent and continuous. Okay. So from here, you can draw your interpretation. Somebody text. Sorry. So your interpretation can be drawn from this, although not many people does it. Oh, my pen is not functioning. I'm trying to activate one moment. I'm trying to activate my pen. It's not working. It's all right. So from this curve, you can divide it. You can divide it depending on how do you want to interpret it. So maybe this is the first phase, this is the second phase, and then this is the third phase. That is the power of having a curve like this. Okay, you can turn mathematical expression and observation into biological discussion and conclusion. So math and science go hand in hand together. Okay, and finally, the fifth parameter that we're talking about is the leaf area. Okay, leaf area is very important again because leaf do most of the photosynthesis. People might argue like, oh, stem looks green, doesn't it do photosynthesis? Yes, it does, but not as much as the leaf. And most of the time, the photosynthesizing stem may be negligible, the product. So usually we don't talk about it because it's negligible. Okay, so for leaf area there is no unit, okay, because it's a ratio. The ratio of the area of your leaf cumulatively over the ground area on which your plant is growing. Okay, so I got this small example here. So the brown is your ground level, the green is your leaf level. So your ground area is 1 square meter, and then your leaf area is square meter. So your leaf area index is 1. It's the same. So your second plant, maybe at more advanced age, maybe 10 days later, while growing on the same area of the ground, it has managed to produce 2 more extra leaves. So instead of having 1, it has got 3 extra leaves. These 3 leaves in total, meaning that you have increased your leaf area index. So your leaf area index is 3, the total area of your leaf area divided by 1 square meter, which is your ground area. So you're going to get your 3. So between this A and B, which one is more efficient? at intercepting the light from the sunlight. This is important, okay, because you're going to have your sunlight here. The sunlight going to come down, and it's going to absorb all the energy from the sun to do the photosynthesis. So that's why leaf area is very important to pay attention to. Okay, depending on the crops, it's going to differ. Okay, some crops, the orientation of the leaf is horizontal. Yeah, like many fruit crops. Yeah, like the mango trees, like tomato, chili, and so on. So usually the leaf area index is around 3 or 4. Okay, because they are horizontal leaf. But for leaves which having the upright leaves, like the rice, you know, the leaf of rice is erect. It goes up, meaning that they can occupy more leaves for a given area. So usually the leaf area is higher for this crop. Okay, so until 6 to 9. And this is the formula. Remember, there is no unit. Okay, even though you see here, actually this is just reiterating the 2 unit of leaf area and also the unit of your ground. Okay, you cancel out, so it become unitless. So this is the leaf area index for a given year of a plant. I forgot what experiment is this, but you can see that leaf area index kind of increasing. Okay, for a plant, and then it's going to become plateau, and then it's going to go down. Why? So in terms of interpretation, if you put like a imaginary line here to turn it into phase, you know that this is the part when the plant is actively growing, the vegetative state, and then this second part here is going to be probably your reproductive, when the plant's no longer producing any more leaf because they are diverting the energy to produce their reproductive structure like fruits and flowers. And then it's going to go down. Maybe the plants, you know, because they are aging and senescing, so the leaves are falling down. So you're not going to get your leaf area index as high compared to when it was much younger. Okay, so since it's in the form of ratio, of course you can convert it leaf area index in the form of percentage. So we got 2 type of plants here, plant A and plant B. So the leaf area for plant A, the coverage only 40%, because you know, if you times this by 10 by 100, you're gonna get 40%. So it looks very, it is very sparse, very relaxed. You can still the ground. Okay, compared to this plant, looks very busy, lots of leaf, meaning that the most of the sunlight that hits this plant from the above is going to be intercepted by the leaf. Okay, so usually in terms of production, usually this kind of plant that we want to look for. Okay, the leaf area is very high. Okay, so this is the leaf area index for throughout the time again. ET stands for economic threshold. This is if you learn your economy letter, meaning that after such point, if your parameter here involved, it will not add any more to the economic value. Okay, but this is something not you cannot control because your plant is keep on growing. But from the economic standpoint, if you can control it, you would ask the plant, okay, that's it, stop it there. Leaf area index at 2.7, because if you increase it anymore, it's not going to give any more profits. Okay, but you cannot do that for a plant. So we can only observe, but we cannot dictate. So all the way until the maturity, the leaf area is going to come down sometime. Yeah. So when you see the word tasseling here, you know that this is the example of a corn plant. Okay, so you can see that the effect of leaf area index throughout the time depending on the density of your crop. So for the high population crop and the very low population crop. Okay, for the high population crop, of course, your leaf area index is going to be very high because you leave very minimum amount of ground exposed, because pretty much your plants are conquering the whole area. Okay, but whether it is economically viable or not, yeah, this is when you use this ET line here, the economic threshold line here. For the high population, you're going to get your economic threshold much earlier compared to other density of population. For the very low population, it never hits the economic threshold. Okay, there is this is what we call here, from here to here. This is what we call as there is room for improvement. Okay, uh, so your plant still have more to give. Maybe you need to increase more of your crops here, or simply you just add more fertilizer. Okay, um, I give you a lot of curve like this because in physiology that's what we're trying to do. We try to understand how the plant function, the mechanism behind it. So if you have something like this, I can teach you easily later how to interpret it. Mathematics and science go hand in hand. Okay, so this is the leaf area index, for example, for three main cereals. We got the sorghum, maize, and rice. We can see that for the circle line here, sorghum is the one which is super high in leaf area index compared to other crops. And now this is the light interception, yeah, of the same plants. You can see that at one point everybody going to look the same when it comes to light interception. Okay, and then if you put everything together, all three crops together, you can actually create an equation in order to predict where they're going to fall. Okay, this is a bit more advanced, but I just want to give you some idea of how useful is it to understand growth analysis and to put it to perspective in terms of your crop growth. Okay, that looks worrying, but not to worry. I just want to show you, even though today you only learn 5 of the growth analysis parameters, actually there are more. So these are the things that people routinely use and routinely utilize when they want to do growth analysis for their crops. There are so many things. Okay, so I give you this. Maybe in the future you want to refer if you have this. So what is significant? For example, if you want to study later the specific leaf area, if you get the leaf area high, what does it mean? Oh, it indicates a less thick and less density of a leaf, or something like that, because that can be translated into a biological explanation side of it. Okay, all right. So toward the end of this lecture, again, please recap about the phases of the plant. We got the germination and seedling, the vegetative, the flowering stage, and the maturity stage. Each of these stage has a way to be quantified in terms of its growth and analysis. Okay, the ones that we have seen today, the 5 things, growth parameters, mainly for the vegetative stage, maybe including some of this germination as well. But for the flowering and maturity stage, usually we use something different. Okay, there are ways to quantify these reproductive organs growth analysis. Okay, just just want, okay, just put this so that again you can see that how the plants grows and develop. Okay, so in your test later, maybe I'm going to put something like this, and I'm going to ask you, show where growth, development, and differentiation has occurred. For example, I mean, why do you say so? I might ask you where differentiation takes the most throughout the course of the plant. Yeah, something like that. Okay. Yeah. So this is just I want to show you the relative time period for a vegetative, reproductive, and ripening for rice. It differs, okay, from plant to plant. Okay. Since rice is an annual plant, this is the time frame that you're gonna get. Okay, so the vegetative phase was going to last around a month or so, and then you're going to get another month for reproduction, and then another month for the ripening. Then you get your rice, and also product it. Okay, I think for today that's all. For the next week, this is what we're going to learn, because this is corresponding to your um, uh, lab practical and activity. Okay. Okay, good. Any question? Yay. Are you clear? Good. No question? No? Yeah, I just want to spend another five minutes or so before you go. We're going to end the lecture now. Do you have class after this, or go back to the bed?
Yes.
Yes.
We have class, Doctor, after
Okay,
this.
okay, got it. That's very fast to answer. Um, can you among yourself decide? I want to divide you into groups. This is for your practical. Okay, I know how it can get if I allow you to find groups during the lab session. It's going to get very busy, very noisy. Okay, I don't want any policemen to come to our lab and summon all of us. Okay, so do the group dividing, group whatever you want to be with your best friend. That's your ex. I don't want it to be in my circle. Decide it before you come to the lab. Okay, so 30 people for each group. So maybe we can have 6 persons for each group. So we're going to have 5 groups for group 1, 5 groups for group 3. Okay, do that. Write your name, your group member names on a piece of paper, and name your group. This is the group that you're going to stick with for the rest of the semester. Okay, for the activity, for maybe the quizzes that we're going to do, the tutorial. Okay, and appoint someone to be the chief of your group. Okay, the name of the group, just put any name. Just don't put any obscene name. I allowed that to student before, did not turn out very good. Okay. Oh, who else cannot come to this lab? Only one student. Anybody else? So just going to 8, 5, 7, 8. Okay. It's fine. All right. Let me think. Just want to double check whether I got everything right. Oh, just to remind you, if you don't like this group or this subject, you can still drop. You got 2 weeks to do so. For the ESMP system, I know the first 2 weeks kind of not so stable. People can drop in, drop out of it very easily. All right. Okay, I think for today that's all. I will try to ask for the lab materials for this week, and then I'll put it on the Putra Blast. Okay. Really? You just want to join now? Okay. All right. I think that's all for today. Any question that you want to ask? You can ask me from earth to heaven. Okay. We because we are learning stuff. So whatever pops in your head, you can just ask. I'm quite okay with that.
Yes, doctor, I have a question.
Yes.
So,
Oh,
just now you talk about the growth measurement, right? So, when
Mm-hmm.
we collect the data for the dry weight, normally how we collect? Because we still let it to be grow continuously, right? So, how we gonna collect the data for the weight?
Oh, so that's why we will grow the plants in many replications. You're going to replicate your plants, okay? Because this is destructive. So you're going to have a lot of seeds, maybe 100 seeds, and then you decide that each week you're going to harvest 10 plants. So we are having assumption here. Since the seeds of the same variety, from the same allotment, from the same of everything, they should behave the same. So even though we have destroyed 10 plants, but we still have the remaining 90 still growing. So that,
So,
yeah.
because every different plant have different growing rate, right? So we assume the hundreds plant are in the same growing rate, is it?
When you get your seeds from the same type, the assumption here, it's got the identical genotype. The genome is the same. When the genome is the same, it behaves similarly, unless if you introduce a different environmental factors. Maybe the plants that you destroy, you grow in highlight, and suddenly the rest of the plants for the next week you dim the light 50%. Of course, they're going to behave differently. Yeah, as long as we use the seeds, the pure seeds. This is why seeds purity very important. The seedlings are going to behave the same way. Yeah.
All right.
Okay.
Thank you, Doctor.
Okay. Okay. Good. People. Nobody want to say anything.
Doctor, can
Yeah.
you share the course outline and the allocation marks for this subject later?
Say again. What?
The course outline and the marks allocation
Yeah, yeah. I, I,
for
I
the
will
coursework.
give that. Yeah.
All right, thank you.
I will put it on the Putra Blast, and then you can download that. Okay, and keep that to yourself. Yeah. Okay. I just want to check with the coordinator of the course. I'm not the coordinator. The coordinator is Prof Uma. I'm going to double check with her. If everyone has agreed, then we'll do that. Yeah. Yeah. Like I said, the first 2 weeks is very unstable, just like you. What, I'm wrong? Okay. Any more questions? Are you good? Okay, I'm going to have another 5 minutes. I'll just let anybody to ask. If nobody asks after 5 minutes, then I think we can dismiss. Okay.
Oh yeah, doctor, how about our attendance?
Attenders. Ooh, good question. Um, I what is this raise hand for the Google? Maybe you want to raise your hand. Maybe that capture as um attendance. Yeah. Oh yeah, 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 yeah. I can use this as your attendance. Yay. Are you okay with my teaching? Am I going too fast? Am I going too slow? Am I not making any sense? Are you this is it not what you're expecting? Or do you want to move to other groups where you can see Prof. Uma and Dr. Phoebe? You know what? It's very interesting because Prof. Umar and Dr. Phoebe was my lecturer like 15 years ago. Yeah, they're very old. But they're very nice people. They're very nice. They're very nice. I mean, like they know me for a very long time. Yeah, yeah. So I think we can make a dismiss at 9:40 if nobody asks make any question. I'll just going to be around. Okay. If you feel like asking, talking to me, just do it. So. This someone just join in. Did you just join in or did you get boot? Okay, 9:40. So no question. I assume that you're happy and then you can go. Okay. So I'll see you on when? Wednesday. Bring your lab coat. Do not wear slippers. Use your pretty gorgeous shoes. Okay. Right. Oh, somebody asked a question here, chat. Oh, sorry. I just uh higher the leaf area, the better. But isn't the greater the number of leaves, the harder for those leaves to capture the sunlight? The, the greater the number of leaves, the harder for those leaves to capture sunlight. Oh, we are talking about the light interception ability. Okay, so you might wonder that if you have too many leaves, it's going to overshadow the leaves at the lower level. Okay, so you have to understand that one thing, when sunshine hits one leaf, not all of the sunlight is going to be utilized by the leaf. Some of it is going to be transmitted. If down the leaf there is nothing else but only ground, the light is going to be lost, just like that. But if you have extra leaf, it's going to increase the chances of the plant to capture as many light as possible. Okay, so this is about getting the full canopy effect. Okay, if it's possible, the plants do not want to allow even one photon of light to meet the ground. It want to capture it all, but of course that's not possible. Okay, so that's why we have some amount of leaf area index. Okay, Good. Okay, any question? Okay, what does it say here? Lost connection, but laptop. Okay, okay. Right. Yeah, yeah. So many issues. Pandai-pandai lah korang. Okay, all good? All good? Okay. Yeah. So if you have more questions, you can simulate it. Okay. And just to remind you about the SOP, please on the day follow the instruction from the lab assistant. Okay. Wear your mask. Okay. Wear your mask. Wear your lab coat. Wear everything that need to be worn. Okay. Do not come not properly attired, okay? Because if you do so, there is a very high chance you are going to be refused to enter the lab, okay? If you for some reason do not have mask, you can borrow from your friends or let me know earlier. Okay, I will ask our lab assistant to provide for you. Okay, not a problem. All right, okay, that's all for today. So I'll see you on Wednesday. Bye.
Thank you, Doctor.
Thank you. Bye.
Thank you,
Thank you,
Doctor.
Doctor.
Thank you, Doctor.
Thank you,
Terima kasih.
Doctor.
Thank you, Doctor.
Ya, sama.
Thank you, Doctor.
Ya, ya.
Thank you, Doctor.
Have a nice day, Doctor.
Ya, you too. Thank you aja. Nasi-nasi tak ada.
Makan-makan PLC, Doctor.
Actually, I don't eat rice.
Oh,
I
you,
teach-
Doctor.
I teach rice science and everything, but I don't eat rice for some reason. And my parents never forced me to eat rice. So sampai dah tua tak makan nasi.
Then what
Excuse
you
me,
eat, Doctor?
Ya,
Doctor.
ya, ya.
This Wednesday I cannot attend the lab as well because I'm in isolation.
Okay.
So Mayani,
What's your metric?
208 397.
208-397. Which group are you? Oh, she lost already. Okay, sorry.
Hello, Doctor. I'm still here. Sorry.
Okay. I just need to know which group are you from? 1 or 3?
Group 1.
Fine.
Should I email letter to you?
Yeah. You can do that. Just so that I have the record. I have already jotted down here because there's the student. Ngu Yiling 208-578. Okay. Tell your other friends if you got problem to come. Let me know ahead of time. Okay.
Okay,
I don't
Doctor.
want to be left out because physiology it can be very tough. So that yeah, you need to stick around as much as you can.
Okay, thank you, Doctor.
Okay, bye. Okay, I'm going to leave now. I'm going to stop recording and then